Chapter 4, Functional Anatomy of Prokaryotes and Eukaryotes. Remember, prokaryotes will include bacteria and archaea whereas eukaryotes will be everything else. So in microbiology, we do focus mostly on bacteria, but we also do need to think about fungi and protozoa are a very significant group when it comes to diseases. So we need to be able to understand how these organisms work. So we need to understand both the anatomy and the function of all the parts for prokaryotes and eukaryotes. On this page, you see an outline of the major concepts in this lecture. Okay. So remember, one of our purposes in this class, why we want to learn about and understand microorganisms, is to identify, treat, or even prevent disease of pathogens, right? But we also want to be able to, you can apply these concepts and this knowledge to learning about the environment and also learning about the beneficial relationship that we have with the microorganisms that live on and in us. So again, remember, this class focuses on disease, but microbiology is so much more than just pathogens. Of course, the pathogens are the super interesting fun part to me and the super important part for you as a healthcare provider. Okay. So in this first section, you want to be able to tell me, what is a prokaryote? What is a eukaryote? What are the three domains of life? And which ones are prokaryotic? Which ones are eukaryotic? Of the eukaryotes, you need to be able to also tell me about some of the kingdoms. You'll also learn about the basic types of shapes and arrangements of bacteria. And so you'll need to be able to look at an image and tell me what kind of shape and what kind of arrangement it is. Okay. So remember that a prokaryote means pre-nucleus, so before or without a nucleus. And both bacteria and archaea bacteria are prokaryotic. So single-celled organisms that do not have a nucleus, and also they do not have any organelles. Okay, if you define an organelle as a compartment with its own membrane, then you can say it doesn't have any organelles. If you don't have that definition, then you would need to state it doesn't have any membrane-bound organelles. Okay. Eukaryotes literally means true nucleus. So these are organisms that have a nucleus. Some are single-celled and some are multicellular. So plants obviously are multicellular. Fungi, yeast are single-celled. Everything else is multicellular. Animals, multicellular. And protozoa are all single or unicellular organisms. Okay. Plants, fungi, animals, protozoa are examples of kingdoms within the domain Eukarya. And there are a lot more kingdoms than these, but these are the ones that we interact with in this class. Okay, so when you're looking underneath a microscope, how would you know if you're looking at a eukaryote or a prokaryote? Well, one thing you could do is look for a nucleus, and if you see a nucleus, you know you have a eukaryotic organism. But another very fast and easy way to distinguish is that eukaryotes are going to be much, much bigger. So prokaryotes are much, much smaller. And I have an image of that later on in this presentation. When we get to the eukaryotes, you'll see that comparison. Okay, so And actually, you'll get to see that comparison in your microscope project as well. So the image that you're going to see later on in this slide, actually probably in part 2, you'll get to do yourself. So that's fun. Okay. Now, most bacteria will have one shape their whole life, and we call that monomorphic. And of course, that's under normal conditions. You could torture them, right? You could add extreme harsh conditions that would cause them to change shape. But under normal conditions, the majority of bacteria will just have one shape their whole life cycle. However, there are some bacteria that are pleomorphic, and that's a very interesting characteristic. Narrows it way down. If you're able to take a sample and see cells in different shapes, and you know that you're working with a pure culture, then you would know that you more than likely have a pleomorphic bacteria that could only be, one of a few types. And the most common example would be H. pylori. So I think your textbook has some images of that you can look at, or you can also Google it to see what does it look like in different stages. Okay, so what is H. pylori? Helicobacter pylori. A lot of people who have stomach ulcers have an infection with H. pylori. But the interesting thing is that not everyone who has the H. pylori infection develops a stomach ulcer, and not every single stomach ulcer has H. pylori. So there definitely is a relationship that seems like sometimes H. pylori can cause stomach ulcers, but there can be other causes. And there can be people that have H. pylori and they're fine. They never developed the ulcer. So we're still learning more about this. Maybe, in another decade, we'll have the answers to this question. Okay, next. Shapes. We call that morphology. A lot of bacteria will be either rod-shaped or a sphere. Sometimes they'll be spiral, and then sometimes there's even other shapes. But these are the most common. Bacillus and coccus are the most predominant types. And sometimes you even get this intermediate, which we call Cocobacillus. And so let's take a look at what does that look like? Unfortunately, before we had better naming system, we had some bacteria that were named Bacillus, so they would be written in italics, capitalized B because it's the genus name. So examples like Bacillus anthracis, which you saw in your first lab, the biosafety lab. And a lot of times in the learning lab, like if we were in person, we would be working with Bacillus subtilis. So in this instance, when we use the word Bacillus, we have to capitalize it and have it in italics because it's a genus name, right? And then other times, if you're just talking about the shape of a bacteria, which you might not even know what the bacteria type is, but you just see it in an image like this, and you would say, oh, okay, it's a Bacillus shape or morphology, right? Furthermore, this is in a chain. You see these long chains. And so we would call that streptobacilli because it's in a chain. So the prefix strepto refers to the arrangement, and that means in a chain. And bacillus, or bacilli for plural, refers to the shape, the rod shape. Okay? If you have an in-between, something that's not completely round and not completely rod, we call it cocobacillus. Otherwise, we call it a bacillus. Now, sometimes you can have these rods can be very small, and they can also be quite large for bacteria, like in the Bacillus subtilis. So you'll see some differences when you look at different images or samples. Furthermore, things like E. coli they don't form a chain, but they just kind of form clusters. And when we talk about bacillus clusters, we call it palisades. So that's the shape is bacillus or bacilli for plural, bacilli. And then here are, if you just have pairs everywhere you look, you see mostly pairs. We call that diplobacilli. If you see long chains as the predominant form, then you call it streptobacilli. And if you mostly just see groups scattered about, then you have palisades. Okay, so here is another example. We can see large rods, right? And we see some that are individual. We see some that are right next to each other. And we even see a small, short little chain, two together. And so what would you call this? Would you call this diplo? Would you call this clustered? Or would you call this chains? Well, I would say that it's mostly just bacilli. No real arrangement observed in this image, because we see everything. We see completely individual. We see like one or two small chains forming, and we see a small cluster. So this could be the start of a strepto. Maybe we were really aggressive when we smeared it, but I wouldn't feel comfortable calling this anything other than just bacillus until I saw more of what was going on here. On the other hand, an image like this, you can see without a doubt this is clusters of spherical bacteria. And so we would call this staphylococci. Staphylo for cluster, cocci for round spherical bacteria. Up here we have maybe cocobacilli, and we see a lot of diplo pairs. So this would be a diplobacillus or a diplococobacillus. Okay. Here we see diplococci, right? We see a lot of pairs, really nice pairs of very spherical. So diplococci. And here we see very nice chains of spherical bacteria. And so that would be streptococci. You wouldn't see anything like this under your microscope. This is fluorescent microscopy. You also won't see anything quite this gorgeous in these images, because these are using advanced techniques, scanning, tunneling, electron microscopes, or just super high resolution. You'll see things that are mostly like this, very small, not as easy to decipher necessarily, but you'll get the hang of it with some practice. Your textbook shows you lots of images, and we skip chapter 3 in your textbook, but chapter 3 is all about staining and other lab techniques. So I really encourage you to read through chapter 3 on your own, because it will really help you to get a better grasp at the lab component, at, steps of staining, looking at like what are the different types of media, common media, growth media, staining techniques, identifying bacteria, all sorts of really useful information. So read chapter 3 when you have the time. And so you might be asking, okay, well, what are these? You can never just look at a bacteria like this and say, oh, that's, Neisseria gonorrhea. You don't know that. You would have to run tests, whether that be a DNA test or a series of biochemical tests. But you don't identify bacteria just by looking at it. Now, in this image, because of this golden color, that is the actual color. In this case, this is not stained. We would know, okay, that's, Streptococcus, like when you have strep throat, Streptococcus aureus for golden. And so I would say more than likely that's what this image is. But again, until you have an actual test to tell you, you don't know. And I would never give you an image of bacteria and ask you what's the genus and species. Rather, I would just ask you what is the morphology, what's the arrangement. Okay, are you using the prefixes correctly? So I'll give you some examples so you can start learning some of the common bacteria. So Neisseria, gonorrhea, and Streptococcus pneumoniae, those are both diplococci bacteria. So it could look like this. Okay, where something like this, the staphylo, you've heard of a staph infection, Staphylococcus aureus. Also, you've heard of MRSA, which is methicillin-resistant Staphylococcus aureus. So we have Staphylococcus aureus bacteria that can live on our skin or in our nasal passages most commonly. And there's about 70% of the population are carriers for this Staphylococcus aureus bacteria in their nasal passages. And then it can get into cuts on the skin and cause an infection. Or it could be spread to surfaces and other people get it, infection with it. And if it gets into the blood, it can become a lethal infection. So it is serious. We take care of that by treating it with antibiotics. But because we've treated it with antibiotics for so long, we've developed, the bacteria has developed resistance. So now we have things like MRSA, methicillin resistant. That's a whole class or category of antibiotics that just won't work against those organisms. We have other types of resistant Staphylococcus aureus as well. So it's becoming harder and harder to be able to treat infections. And unfortunately, we already have a large amount of people die every year from resistant infections. We just don't have the drugs to treat them, or by the time we learn that it's resistant and the type of drug to treat it with, the person may have already died from the infection. So this is one of those hot topics. It really should be getting a lot more publicity so that people understand how important it is to take their antibiotics correctly, follow the instructions, and not to overuse them and take them for granted. Okay, next example, the streptobacillus that we saw here, streptobacillus, an example of an organism that causes disease, a Streptobacillus monoformis can cause what we call rat bite fever. So not the most common disease, but it is an example. So there you go. You have an example of Staphylococcus bacteria, Staphylococcus aureus. You have an example of Streptococcus pyogenes, which can cause strep throat. Streptococcus pyogenes can cause strep throat. Staphylococcus aureus can cause skin infections, and that can get into the blood, cause systemic infection. And diplococci could be something from a STD like gonorrhea, or pneumonia, Streptococcus pneumonia. There's a few different species of Neisseria. If it's gonorrhea, then it's the STD, but there's also a form that we'll learn about later on in this class when we do our discussion on diseases. So again, I bring up these different examples because I want you to start getting used to hearing these names and associating some of their traits, so they'll be easier, a little bit easier when we go through our overview of diseases. Okay, so arrangement, a little bit more about how do bacteria make these different types of arrangements, and it has to do with how they divide during replication and division. So if it divides always on one plane, then you're going to get diplococci, and if they stay attached while they continue to divide, then you'll get the formation of streptococcus. However, if it has two planes of division where sometimes it divides one way, sometimes it divides the other way, then you can get the formation of a tetrad, which is really cool, like the square, right? 4 cells. If that continues to divide while they're attached, then you can get to this cube, okay? And that cube can continue to grow and grow up, even to 64 cells. And that will give you a very nice square shape. You will not really see that in our class, but you will see a lot of streptococcus, you'll see a lot of diplococcus, occasionally a tetrad, and then a lot of these clusters where there's several planes. It divides every which way, and it forms like grape-like clusters, which is where we get the name Staphylo from. And it's circular, so Staphylococcus. And as it continues to grow and divide, it just becomes this bunch, right? So that's what we mean by a plane of division. Single plane, single line. 2, you get the squares, and several planes, you get the clusters. Okay, so you don't always have circular or rod. Sometimes you have these spiral or even curved bacteria. So Vibrio looks kind of like macaroni, right? And notice it also has a single flagella coming out at the end. This is a scanning electron micrograph, so you're getting a very high resolution and you can actually see the flagella. You would not be able to do this with the regular microscopes that we have in lab. Okay, same with all of these. We're getting really high resolution so you can see the detail. So something like this is thick and very curved. This is called a spirillum when it's one, and spirilia if there were more than one. And it's rigid, it's thick, and it can have flagella at one or both ends, depending on the species. A common example is Campylobacter. Campylobacter is common in chickens, and it can cause a very serious intestinal infection if you eat food that has Campylobacter in it. This is more common in the UK, where they eat pate, which is not completely cooked meat. Okay, spirochete is a very thin spiral bacteria, and it moves in a corkscrew motion. So looks very different from spirillum. Okay, you would not confuse these two. These don't have flagella that stick out on the ends. Instead, you can think of it as having a flagella that wraps all the way around. When it wraps all the way around, it's no longer called a flagella, rather it's called an axial filament. And I'm going to show you a little bit more about that in a moment. But for now, just take my word for it. This is a very flexible cell. It moves, the cell itself moves a lot. So this stays intact as is, and the flagella just moves it around. Whereas this actually constantly changes its shape of its body to move. So this is like kind of worm-like, whereas this is just being propelled, moved around. Okay. Common examples of spirochetes, Lyme disease, Borrelia burgdorferi, and Treponema pallidum, which causes syphilis. Did I have more I want to tell you? No. Occasionally I have notes in my slides, but I already told you everything that's in there. Okay, now I'm showing you this because it's really cool. It's really fun, right? Bacteria can exist in all shapes and sizes. Well, not all shapes, not all sizes, but many types of shapes. So some cool ones are these rectangular ones that this is actually growing on a crystal of salt. And this one is a star-shaped. This always makes me think of gushers. I don't know if you guys know what those are, if it still exists, but when I was a kid, there was this weird like candy called gushers. And it always reminds me of this. But isn't this neat, seeing these different shapes, and just there's such a diverse world out there. We only get such a tiny view of it in our courses, but the world of microbiology is amazing. Okay, so these two examples of prokaryotes are actually Archaeobacteria, I believe, because they're living in extreme environments, right? Like on salt as a halophile. And this one, I think it's in the ocean, but I'm not even sure where the star-shaped bacteria is coming from. So we don't get to work with these in this class. This is the only time you'll see it. All right, so now we're going to look at structures external to the cell wall, which we kind of already started to talk about a little bit with talking about flagella and axial filaments. So we're going to take a closer look at that. And also, we're going to look at something called the Glycocalyx, which is on top of the cell wall. So these are external structures. Okay, so some diplococci, such as Streptococcus pneumoniae, which causes pneumococcal pneumonia, which we do have a vaccine for, and you should be vaccinated for this. Make sure you get your vaccinations. Anyways, so not only do we see a pair, which is diplococci, we also see this huge barrier surrounding it. The cell wall itself is this thin layer that you see. The thick yellow portion is what we call a capsule. And a capsule is one of the types of a glycocalyx. Okay, so glyco means sugar, right? You think of glucose or glycogen, right? So glyco for sugar and calyx for coat. So we have this sugary coating, this carbohydrate coating, and there's some proteins in there too, and even some lipids, but it's mostly carbohydrate. And the cell is secreting this material and building this, so it's secreted by the cell. And if it's what we call neatly organized, very tightly, neatly organized, it's called a capsule. So this forms a pretty smooth edge, right? So this is a capsule, and capsules give a lot of protection. I don't know why this is showing here. Please ignore that. But capsules give a lot of protection to bacteria. They keep the bacteria from drying out when they're on surfaces. and they can help protect it from our immune system if it's in the body. So we call it an invisibility cloak because our immune system has a really hard time being able to do anything with a capsule like this. All of the surface antigens, or the majority of the surface antigens, are being covered up. So if we cannot access the surface of the cell wall, then we lose a lot of our strategies for fighting. So, invisibility cloak. Now, some bacteria secrete this glycocalyx, but it's not so tight. It's not forming this smooth edge. And then we call that a slime layer. So, unorganized and loose slime layer. And why would a bacteria want a slime layer? Well, it still gives it some protection from, drying out, but it also allows it to attach to a surface and to each other and form colonies. And one of particular importance is a biofilm. Biofilms are especially problematic. We don't have any good method to deal with biofilms. They're basically impenetrable. We can't get through it. We don't know how to get rid of it. It is really common for biofilms to form when there's moist environments. So like on a filter, in water treatment plants, and on teeth. If you look in your book, you'll see an image of some very unhealthy teeth with biofilms on them. Okay. And in hospitals, this is a huge problem whenever we insert something into someone's body. So whether it be a urinary catheter or a pacemaker or anything else that's not actually flesh tissue, you have now provided a surface that bacteria can attach to and form biofilms. Not all bacteria is capable of making a biofilm. Only some species are, right? They have to have the genetics that give it the ability to make and secrete the glycocalyx. But even still, there's enough different types. This actually amounts to being a huge problem in hospitals and water treatment plants and dentists for our teeth. Okay, so biofilms are very resistant to chemicals, to antibiotics, and to the immune system. So we just don't really know how to get rid of them once they form. So if a patient had a biofilm forming on their pacemaker, on their catheters, or whatever, you'd have to remove it and replace it with something sterile and hope that you don't get another biofilm forming on it. Okay, so I started talking about flagella a little bit earlier, and now we're going to take a little closer look at that. So why is it called flagella? The type of protein that it's made out of, chains and chains, repeating units of this protein called flagellin. And this is used for motility. Some bacteria don't have flagella at all. Others have one, and others have more than one. And there are different arrangements. So you'll see something like this guy. I call him Perry Hairy. He has flagella all over. He's hairy. So Perry Hairy, that's how I remember Peritrichus, right? All over. You might already know the root peri as being multiple or all over, right? You might know what this means already, but for those of you trying to remember, like I once did when I was a student, you can remember Perry Hairy. Amphi means both ends. So Amphitrichus would mean it has one or a bundle at each end. And actually, Amphitrichus should mean tuft at both ends. Lophotrichus means a tuft at only one end. And monotrichus means it has only one flagella. And of course, putting the A in front, atrichous means no flagella at all, and we don't even usually bother to say that. This is a super cool image. It's fluorescence, right? Staining different proteins so you can get this nice contrast. And of course, the green is showing you all the flagellin protein. They also mentioned in this description that this guy is from the genus Proteus, and that he's capable of a type of motility called swarming. They also give you a fun fact that some peritrichous bacteria can have more than 1,000 flagella, which is that's just incredible, right? That's so cool. So swarming is the most advanced form, the highest level of motility, and we'll take a look at that in just a moment. So here are those arrangements I told you about. So perihairy has flagella all over, so peritrichous. Mono and polar means just a single flagellum at one end. Amphitrichous and polar means two, and the two are at the ends. And then lophotrichous and polar means a tuft. And yeah, that's pretty much all I have to say about that. It's just putting some images to these terms. Make sure you memorize them. Okay, so I wanted to talk to you about swarming. I gave a lot of images just because it's so cool, guys. This is so gorgeous. Like this is one of the reasons you go into microbiology, because this is just so neat, so cool. Okay, so you're not going to learn anything really too specific about these images, but just want you to see, oh, how pretty. Okay, but we are going to learn a little bit about swarming motility. So I told you it's the highest, most advanced form of motility, and it is. And it actually provides a lot of advantages to the bacteria if they're able to do that. They're able to move around very rapidly. They're going to have better access to get nutrients, get away from something harmful. And it also just, there turns out that bacteria that have this ability tend to also be highly resistant to antibiotics, and that can be for lots of reasons. And we also see that they're able to escape, get away from being eaten, like an amoeba, for example, likes to eat bacteria, and they're not going to be fast enough to get their hands on, well, they're not real hands, pseudopods, but amoeba are not going to be able to engulf swarming bacteria that are able to swarm. The bacteria will get away too fast. Okay, so this gives a huge competitive advantage to bacteria that have this. We also see that they have these bacteria that swarm secrete special substances called surfactants, which are like kind of slippery, kind of soapy. And the surfactants themselves might offer them protection. In order to have swarming ability, you need to be able to have lots of flagella, right? And you need to have flagella-mediated surface migration. So the ability to have this feedback with knowing you're on a surface, that it's time that you can implement swarming. So bacteria will not instantly swarm. There will be what we call a lag period where they make the surfactants and all the things that are involved in being able to ramp up to swarming. So here we see whenever we test for swarming, you put a sample in the middle, and then you incubate it, and you see if over time if it moves away from the middle. Here we see this really cool bullseye pattern, where the bacteria have migrated, they grow in these dense rings, and then they migrate, and then they grow in a dense ring, and they keep doing that pattern. And here we see the bacteria did not move away from the center. So this is non-motile. This here may or may not be motile. We see some growth here, and we would need to confirm that that's actual motility or not, because it's possible that this was just condensation that allowed these bacteria to slide around on the Petri plate. So I would not feel comfortable saying what this is without a confirmation test. But everything else, you can see tons and tons of bacteria moved away from the center. Just like in here, this is the same organism, Pseudomonas aeruginosa, on every single one of these plates, giving it more and more time. We have samples of just what we call wild type bacteria, means the bacteria you find in nature. Pseudomonas aeruginosa has become problematic in hospitals. We'll be talking about this guy in the future, okay? But I want you to just appreciate how awesome this is. And remember, when you're testing for swarming motility, you have to put your sample just in the middle, incubate it, check on it for a few days in a row, up to 10 days and max. I mean, these only took 24 hours, so that's real fast. Sometimes motility can take a week or so to form, and you want to see if it can move away from the center. All right, and this is just more patterns. This is an organism called P. Vortex. It's just beautiful, cool pattern. And the same organism in each of these images. So that's really fascinating. There's a lot that we still don't know, that we still want to study when it comes to the swarming motility. And so A through D, A, B, C, and D is this P vortex. And then the bottom left is Bacillus subtilis. So this one's Bacillus subtilis. Everything else is P vortex. But yeah, just super cool. I'm not going to give you an image and ask you what the name of the organism is. I just wanted to share this with you. Okay, so I do want you to learn a little bit more about the type of movement that bacteria can do when they have flagella. All of the flagella have to be moving in the same direction at the same time. So they'll bundle together and they'll move in the counterclockwise direction, and that will propel the bacteria forward. If one or more of the flagella starts to rotate in the clockwise direction, it'll cause all of the flagella to separate, and that will cause the bacteria to tumble. Whenever the flagella start to all go in the counterclockwise direction again, it will propel it forward. And this will be completely random. So it's a random movement. Well, maybe I shouldn't say completely random. So why would it suddenly stop moving forward? It gets some chemical cue. And so we call this chemotaxis. It might be trying to move towards some food or away from something harmful. And so when you see a lot of tumbles, we call kind of a random, it's not really doing anything in particular. But when you see longer runs and fewer tumbles, then we know chemotaxis is happening, and we call it kind of a biased random walk. Because the bacteria doesn't have fine control to steer continuously in a particular direction, but it can go for longer runs when it's going towards an attractant that it's trying to get to. So here's a short video for you that will describe what I just said, but you'll get to see it in motion. Make sure you pay attention to the details here. And right, so counterclockwise, it propels forward. Clockwise, it will tumble. All right. Here I just wanted to show you, so gram-positive has a plasma membrane, and it has on top of the plasma membrane, it has a cell wall, and the flagella will attach to the membrane and go through the cell wall, and that's what it looks like. So here's the closer view where it's anchored into the membrane. It goes through the cell wall, and then you have the filament. I'm not going to test you on the mechanics of the flagella. It's super cool, but we don't have time for that. In gram-negative, I want to show you, because gram-negative have two membranes, an outer membrane and a plasma membrane. It actually has this anchorage, just like you saw in the previous image, but then it has this second set of anchorage in the cell wall and the second membrane. So it's just a little bit different looking. But as far as the part that goes outside the cell, looks exactly the same. Mostly just want to show you this image, introduce you to the idea of gram-positive, gram-negative, and the difference in their cell structure, which we're going to look at. So lastly, I mentioned axial filaments earlier, and now you can see there's a thin layer that covers the filament. The filament is on top of the cell wall. So you have the cell wall, then the filament wraps around it, and then you have an outer sheath that goes on top of that. And this is how things like the organism that can cause syphilis and the organism that can cause Lyme disease, this is how they move. It's really cool. Okay, so spirochetes, axial filaments. It's anchored at one end, wraps around, and then rotation of the filament will cause the entire organism to move and propel. Okay, so go ahead and read about that in your textbook, and also here's a short little clip to show you what it looks like under the microscope. Fimbriae and Pili. Fimbriae are what I like to call fingers. Fimbriae, fingers. They're for attachment and attachment only. They do not provide any motility. Attachment's important for forming colonies, like you would see in infection or biofilm formation. Another structure external to the cell wall is what we call pili. A single one is called a pilus. These are very long and thick structures, right? Whereas the Fimbriae are very short, okay, and thin. Pili play a very important role in that they can, one bacterium can transfer DNA to another. And this is why antibiotic resistance can spread so quickly, because you don't have to wait for the bacteria to divide and multiply. They can start spreading it all around. This process of giving DNA from one organism, from one bacterial cell to the next, is called conjugation. Sometimes bacteria can also use their pili to grab onto a surface, pull themselves towards it, and release and repeat. So it's this very chug, chug, chug kind of, motion, which we could call like twitching or grappling hook like motility. The main function for pili is conjugation. So make sure that up to this point you've been following along, understanding and remembering. So what's the significance of bacterial capsules? What are different ways that bacteria can move? Can you name all the types and examples? And would you be able to look at some bacteria under a microscope and tell me their shape and arrangement? Okay, so I think this is a good stopping point for the first video. We'll probably end up having to make two, one to finish up prokaryotes and then one for eukaryotes. All right.